Okay, good afternoon, everybody. The Biochemical Society and Portland Press are pleased to welcome you to this webinar, which is part of our biochemistry focused webinar series. Topics in the series include different research areas in the molecular biosciences, as well as practical sessions to support career development. Each webinar will give you the opportunity to ask questions via text, and we welcome suggestions for future topics and speakers to feature in our webinar series. Please see the website for more details. I'm Hannah. I'm a postdoctoral research fellow based at the Centre for Medicines Discovery at the University of Oxford. And my research is focused on the role of the ubiquitin system in mitophagy in relation to neurodegeneration. So I'm very much looking forward to today's webinar, which is titled The Role of Ubiquitin from Ring Domains to Mitochondrial Inheritance. It is part of our dedicated early career research programme of webinars, and we are delighted to provide this opportunity today for Jane and Michele to share their work with the molecular biosciences community. Before I hand over to our first speaker, I'd like to mention that questions will be asked at the end of the webinar, but please do send in your questions during the talks. If you have a question, please type it in the question box as shown in the image on the screen, stating who your question is for, and we will try to answer as many as possible at the end. So today, our first invited speaker is Jane Dudley from the Francis Crick Institute. Jane graduated with a first-class biochemistry degree from UCL before moving to the University of Edinburgh for her PhD, where she uncovered an interplay between the cellular recycling pathway, autophagy, and oncogenic signaling in the context of brain cancer. Following 2 years working with research funding agencies, she is delighted to have made the move back to academia in Katrin Rittinger's group at the Francis Crick Institute, employing a multidisciplinary approach to explore the biochemical mechanisms, structural properties, and cellular functions of ubiquitin signaling. Today, she will be presenting her work on discovering pseudoenzyme structural variants in ring ubiquitin ligase domains. So I can see your screen, Jane, so it's over to you.
Brilliant, thank you very much. Thank you very much for having me, and thank you everyone for coming along on a Friday afternoon on a hot day. I know it's not easy to find the time. And thank you to the Biochemical Society for giving me an opportunity to talk today about my work looking at ubiquitin ligases. So I'm aware that some people might be coming at this without much of a ubiquitin system knowledge, so I'll just introduce the ubiquitin cycle quickly. So ubiquitin is a small protein which can post-translationally modify substrates, and this is mediated by a cascade of E1, E2, and E3 enzymes. And then the ubiquitin mark can be removed by deubiquitinases or DUBs. And we're particularly interested in the E3 enzymes. So these are the ones that specify the substrate. So they bind the substrate and dictate when and where substrates are ubiquitinated. And we think it's really interesting to understand that there's about 670 E3 ligases, but there are over 20,000 proteins in the proteome. So understanding when and where ubiquitination happens is a real focus for the Rittinger lab. And it's something we're trying to get to the bottom of in this talk as well. So protein ubiquitination can result in a lot of different outcomes in the cell. Most famously, it can result in degradation of substrates either via the proteasome or via the autophagy pathway. And this has really important implications in the expanding field of targeted protein degradation, where E3 ligases can be synthetically linked to either substrates they're already linked to or to novel substrates. So for example, if you're thinking about oncogenic proteins, you can target them for degradation synthetically using small molecules. But it's not just in degradation where ubiquitination is important. It also is important in signal transduction. So your proteins can be modified and change how the signaling happens through the cell. Ubiquitination of histones can modulate transcription, and basically any pathway you think of, if your protein can be ubiquitinated, your pathway can be modulated. So ubiquitin is really important all over the cell. And we're going to be talking today about the TRIM protein family. So these are called TRIMs because they have this characteristic tripartite motif of a ring domain, one or two B-box domains, and then a coiled coil domain. And then at C-terminus, there are these kind of variable domains, and they classify the TRIMs into these 11 different classes. So the C-terminal domain is thought to mediate substrate binding. The coiled coil mediates homodimerization of TRIMs. The B-boxes regulate either multimerization or in some cases autoinhibition, and in some cases it's not known what they do. And then at the N-terminus here, you have this ring domain. And this is really where it was assumed that all of the TRIM proteins would be ubiquitin E3 ligases, where they would bind the substrate and then the ring domain would confer ubiquitination activity and ubiquitinate the substrates. However, ourselves and others had kind of anecdotally seen that some of the TRIMs seem to be unable to catalyze ubiquitination. But it was kind of difficult to compare because a lot of the studies were done in different conditions, different cell lines, in cells, in vitro, and that's kind of where my project started. So we want to understand throughout the whole family which TRIMs actually do have ubiquitin ligase activity and which ones don't. So I'm going to talk about half of the screen that we use to address this today. And this is this kind of in vitro ubiquitination screen. So to do this, I developed a panel of all of the TRIMs with GFP tags, and these were then transfected into HEC293 T cells. The GFP was IP'd, and then this GFP tag TRIM was mixed in, and the in vitro ubiquitination cascade was reconstituted. So we have then recombinant ubiquitin, E1, and a mix of different E2 enzymes. And this mix of E2 enzymes was designed to try and cover the most common catalytic mechanisms that are used by rings. So most of these E2s have been shown to work with ring domains before. After this reaction has happened, I then analysed how much ubiquitination had happened using an analyzer that's specific for conjugated ubiquitin. And this gave us quite a few what I was calling hits, and these are TRIM proteins that lack ubiquitination activity. And these are the ones I'm going to be following up on in the rest of the talk. To understand what was going wrong in those particular cases, we need to understand more about how ring domains actually work. How do they catalyze ubiquitination? So unlike hex domains, for example, which have a catalytic cysteine and bind the ubiquitin directly, ring domains actually work by kind of so-called scaffolding the E2 ubiquitin. So the ubiquitin bound by a thioester bond to the E2 enzyme, the ubiquitin is quite floppy and it's so-called open conformation. But when a dimer of ring domains bind, it holds the E2 ubiquitin in this closed conformation, which strains the thioester bond for attack by a substrate lysine and discharge then of the ubiquitin onto the substrate. And to do this, as you can see in my diagram here, you need to have two ring domains because they both contact the ubiquitin. And these contacts are needed to hold this ubiquitin in this closed conformation. And each of these monomers contacts an E2 enzyme. And you can see this quite nicely illustrated in a crystal structure solved by our lab a couple of years ago. You can see in the different shades of orange, two ring domains in a dimer from TRIM2. In magenta, we've got the E2 enzyme, and the ubiquitin is in teal. And you can see here at the top, we have this 4-helix bundle, which mediates the dimerization. And down here, we have what I'm calling the core of the ring domain, and this can contact the E2 and the ubiquitin. In particular, there's this so-called linchpin residue in the ring domain in orange here, which you can see contacts both the ubiquitin and the E2. And these contacts are known to hold this ubiquitin in this closed conformation so that it can be discharged onto substrates. And therefore, this is the kind of the core of how the ring domain catalyzes ubiquitination. And so our inactive TRIMs come in two different flavors. Those that have disruptions in the 4-helix bundle, and so are monomeric rather than dimeric, and those that have disrupted mutations in the core of the ring domain, meaning that they're unable to properly contact the E2 ubiquitin. So I'm going to talk about the ones that have disruptions in the 4-helix bundle first, and so therefore are monomeric. So if we look at this kind of characteristic TRIM2 active TRIM ring domain, you can see these nice helices at the top. If we look at TRIM24, 28, and 33, we can see that although there's some propensity for helices to form here, largely they're disordered regions here, and this is just predictions from AlphaFold. So we would predict, therefore, that these are unable to form this 4-helix bundle and therefore unable to dimerize. And we tested this using SEC-MALS. So this is size exclusion chromatography linked to multi-angle laser light scattering. And this can measure the size of the molecule and that you're putting through it and in solution. And for us, this is really important because we can then look at the oligomerization state of the ring domains. And so, for example, for TRIM28, we can then see that this is strictly monomeric, so it doesn't form these dimers. And this correlates with this lack of activity. So we've got our positive control here, TRIM25, and when we undertake a ubiquitination reaction, you can see these nice smears of ubiquitin, which indicate these kind of ubiquitin chains of high molecular weight that get formed. Whereas for TRIM28, we can see that there's nothing really going on there. No matter which E2 enzymes we try to work it with, there's no activity. And this is a similar picture for TRIM3. So for TRIM2, we can see this nice ubiquitin smear. TRIM3, we can't see anything at all, it's inactive. And similarly, we can see that this N-terminal helix doesn't form properly in TRIM3. TRIM15 as well, we see that it has these unfolded N and C terminal helices on the ring domain, as predicted by AlphaFold. Obviously, pinch of salt, we had to prove then that this kind of lack of helices was correct. This then correlates again with this lack of activity for TRIM15. To kind of support this prediction that we didn't make crystal structures, but we could look at NMR to see how much of the protein was disordered. And although there was some supporting evidence that there was quite a lot of disorder in TRIM15, actually it suggests that there was probably a bit more order in the core of the ring domain than there was suggested in the AlphaFold structure. So possibly it's closer to this Rosetta fold 2 structure prediction. So as I was saying, these helices are thought to mediate dimerization of the rings. So for TRIM2, our active TRIM, we can see it exists in a monomer-dimer equilibrium. Whereas for TRIM3 and TRIM15, the ones with disrupted helices, we can see that by secmoles again, that these are strictly monomeric. So that gives us our first class of ring domain that's inactive. And the second ones are ones that seem to have lack of proper contacts with the E2 ubiquitin. And this kind of threw us at first because we did secmars again on some inactive TRIM rings, so TRIM6 and TRIM22. We said, oh gosh, but they're actually dimeric. So how is it that they're inactive? So we then started scratching our heads, and thank God for AlphaFold 2 really pointed us in the direction we needed to be looking. We could see that actually TRIM6 and TRIM22 have this glutamine in the position of the linchpin, so-called linchpin arginine, that's seen in active TRIMs. And also in TRIM6, we can see that this helix here seems to be slightly unfolded. And interestingly, in TRIM22, although it can form dimers, it has a bit more of a propensity to be monomeric. And we think this is because of these 2 lysines that are actually pointing at each other in these helices at the top of the ring domain, and therefore potentially disrupting how stable that 4-helix bundle is. So we had our hypotheses about why these weren't going to be active. And quite satisfyingly, if I made a bunch of point mutations that correct these mutations in the ring domains of TRIM6 and TRIM22, we can restore activity to a similar level to our positive control. And really, to try and answer whether this was to do with its interaction with the E2 ubiquitin conjugate, what we did some NMR. And so we had labeled E2 enzyme, which is what you're seeing here in the ribbon structure. And what we would expect to see if we titrated in a ring domain that would be active is what you'd see for this active TRIM6 mutant that I made. You can see all of these chemical shift perturbations across the front and the back of the enzyme as the ring domain allosterically activates the E2. But what we see in the wild type ring domains is actually just a few very weak perturbations in residues, mostly at the front, so-called front side of the E2 enzyme, showing that the TRIM6 really isn't activating the E2 properly, that in such a way that it would be capable of discharging the ubiquitin. And my final example I have for you is TRIM51. So I've aligned it here, its structural prediction, I've aligned it with TRIM49, which is extremely closely related to TRIM51, as I'll go into in a minute. And you can see in TRIM49, it has these nicely folded beta sheets, or predicted to have these nicely folded beta sheets, and quite bulky aromatic residues down here. Whereas for TRIM51, it's quite a lot smaller residues here. These beta sheets aren't predicted with any confidence at all. And we also have a charge change here to these 2 arginines as well. So you can see it's quite different structurally there. And this results in changes in its ubiquitination capability. So TRIM49 we can see is nice and active, TRIM51 is not. And quite nicely, if we swap these 2 regions between these proteins, we can restore activity to some extent for TRIM51 as well. So really kind of pinning it down to this lack of activity due to this kind of folding here. And so after finding all of these different TRIMs that seem to lack activity, I then dove into literature and found this whole pseudoenzyme field, which is really well established, in particular for pseudokinases. So they're enzymes that have really high sequence homology to the active forms of the enzyme, but seem to lack activity. And for this case of pseudokinases, they've been really well described to either activate or inhibit the active kinases. So they're a really important regulatory mechanism against the active forms of the enzyme. And so this is what we've got here. This is exactly the same. We've got these proteins that look just like the active enzymes, but they have these key mutations that mean that they can't catalyze ubiquitination. So I call them pseudoligases. And I thought it'd be quite fun today to kind of take a step back and think about what it is that these pseudoligases are doing. So we found them, and now we need to understand what it is that they actually do. So active TRIMs are found in all multicellular eukaryotes, but these pseudoligases seem to have emerged at different times through evolution. So for example, TRIM15, 24, 28, and 33 seem to be found in quite a lot of other species in their kind of inactive form, whereas TRIM3, we only find that in placental mammals in its inactive form. So at some point, the gene has duplicated, and in some species, it's gone on and been active TRIM3, and in placental mammals, it seems to have lost activity. For what reason, we don't know. And TRIM51 it's just specific to humans. TRIM22 and TRIM6, these genes are specifically only found in primates in their inactive form. So to me, this says that at some point through evolution, these TRIMs have duplicated, and for some reason when they've lost their activity, this might have given a selective advantage in evolution as well. So kind of hints that there's some kind of function for these TRIMs, I think. And I can give you two examples now of places we think that these TRIMs actually have a function. So TRIM2 and TRIM3 are homologs, and TRIM49 and TRIM51 are homologs, and they have really high sequence identity, whereas TRIM2 is active, TRIM3 is not, TRIM49 is active, TRIM51 is not. So they kind of seem to come in these pairs where they've got this kind of high sequence identity, and one's active and one's not. And what we found actually is that you can see TRIM3 here, you can see that these residues have changed specifically in placental mammals, whereas in other species, you can see that they actually still have residues that would be conducive to forming these helices, and therefore dimeric and active. And we can see that TRIM2 and TRIM3, they co-IP, so they pull each other down and interact in cells. And the presence of TRIM3 represses the activity of TRIM2. So it seems that TRIM3 has some kind of negative regulatory action against TRIM2. And this is paralleled in TRIM49 and TRIM51. So they can co-IP each other, and the presence of TRIM51 represses the activity of TRIM49. And we think this might have some functional impact. We see that TRIM49 overexpression increases autophagic flux. So you can see here increased LC3 lipidation, and TRIM49 co-localizes really nicely with LC3 in cells, whereas TRIM51 is really diffuse in cells, doesn't co-localize with LC3, and seems to actually repress LC3 lipidation to some extent as well. And this mirrors some results that were actually seen in a screen from Boho Deretich a few years ago, where he screened quite a few of the TRIM family proteins for impact on autophagic flux. So kind of interesting that we were able to pick that up and maybe provide some kind of explanation about why they might be having opposing roles. So I'm a bit ahead of time, but to summarize, approximately 12% of TRIMs seem to be these pseudoligases that are unable to catalyze ubiquitination. And this is because there are really small sequence changes within the TRIM ring domains that cause these structural perturbances at really critical interfaces that disrupt its ability to either dimerize or bind the E2 ubiquitin, and together both of those types of mutations result in the inability to hold the E2 ubiquitin in this closed conformation and discharge ubiquitin. And what I think is really interesting, kind of thinking about the functional side of this, is that they seem to have different roles. So TRIMs 24, 28, and 33 have been really strongly implicated in regulating transcription and are implicated in different cancers. But they're not to do, clearly from our studies, we can show that that's not to do with their ability to add ubiquitin to substrates. There have been some reports that they might add as sumoligases. So this is when you add small ubiquitin-like modifier to proteins. Or I think what's quite a cool idea is that maybe they're just acting as protein-protein interaction scaffolds. As I was saying at the beginning when I introduced the domain structure, really what you have is the substrate binding domain at the C-terminus. You have a cord-cord domain, B-box domain, and then the ring domain. And what all of those are doing is they're all carrying out protein-protein interactions of different kinds. And so really might the trim have been, if it's inactive, might it be co-opted to act as a protein-protein interaction scaffold for a larger complex, then it doesn't ubiquitinate, just kind of holds in place. I think that's quite a tempting idea. I've been able to show you that for a couple of them, they seem to, these pseudoligases seem to inhibit the active forms. And what is actually really exciting is that for some of them, we just don't know. So for example, for TRIM6, it's been very convincingly shown that TRIM6 can restrict viral replication. But if it's not by ubiquitination, then what is the mechanism? And can we actually take advantage of that therapeutically? So there's a lot more work to be done. I'm currently, I just thought a bit of a sidebar at the end. I'm currently kind of moving on from this paper, which we published earlier this year. And I'm now trying to understand a bit more about how TRIM ligases work functionally. And to do this, I'm focusing on the brain and using some cell models of the brain to look at how TRIM biochemistry translates to physiological roles in neuronal development and also in brain cancer. And this is something I want to focus on as I start to take myself further towards being independent, and I'd like to start my own group along those lines. So it just remains for me to thank my lab. A huge thank you first to Katrin Rittinger, who has been a fantastic mentor and has given me the space to follow the data and really take this project to completion and find this whole new class of pseudoligases. Her support's been amazing. Thank you very much to Diego from our lab, who's really a structural wizard and helped me kind of find my way through some of these techniques, particularly the NMR. Big thanks to all the services and platforms at the CRIC who have been a fantastic collaboration throughout. Big thanks to our collaborators Ian Taylor and also to the Teresa Thurston's lab, originally at Imperial and now moved to Oxford, who started us off with some of these reagents as well. This QR can take you to the paper if you want it. This is PubMed ID, and I look forward to your questions after the next session. Thank you.
Thank you, Jane. That was a really interesting talk, and I'm looking forward to the question session at the end. So moving on, we have our second invited speaker, who is Michele Frisson from the University of Cambridge. Michele is a cellular biologist with an eye on the role of the science and the scientist within society. He graduated with a first-class biochemistry degree from UCL in 2013 and directly moved on to undertake a London interdisciplinary doctoral programme within the lab of Michelangelo Campanella at the Royal Veterinary College. After completing his PhD in 2017, he then moved to the MRC Mitochondrial Biology Unit in Cambridge to work in the lab of Patrick Chinnery. In 2020, he assembled the most environmentally conscious MVU members to form the Green Team, interfacing the MVU administration to reduce the Institute's environmental impact. He has since remained chair, leading the Green Team to win several awards and become one of the most proactive institutes within the MRC and the University of Cambridge. Sorry about that, there's some building work going on here. Today, he'll be presenting his work on how ubiquitin-mediated mitophagy regulates the inheritance of mitochondrial DNA mutations. So I can see your screen, Michele, so it's over to you. Thank you.
Fantastic. Thank you very much. What a wonderful introduction. Yeah, it's great that actually the importance of the ubiquitin-proteasome system has been talked about. It's great to have such a wonderful chair who's interested in mitophagy and a presentation about the role of evolution in these processes. Because really my talk is about that, and to introduce it best, I think I have to talk about the concept that eukaryotic life is made possible by the coexistence of two genomes. The nuclear genome exists in two copies. It's much larger than the mitochondrial genome, but the two copies, one is inherited from the father, one is inherited from the mother. While the mitochondrial genome is much smaller, is present in many copies, and is inherited strictly maternally. The mitochondrial genome mutates at a much faster rate than the nuclear genome. If we look, for example, this is a sort of phylogenetic plot where the mutational rates of the nuclear genome over the mitochondrial over the nuclear genome are shown, different groups of species. And what we can see is that this ratio is always higher. showing that the mitochondria mutates faster than the nucleus in all of these species. But this can range from sort of 5 to 10 times faster to all the way in mammals to 10 to 20 or even 40 times faster. But this mutational rate is not neutral. And actually, in many cases, it is very impactful towards evolution and towards human health. So this is a recent study where 2 sister species of fish were crossbred. As I said, the mitochondrial genome is only inherited maternally, so there's only one type inherited, while the nuclear genome is inherited from both parents. And by hybridizing these fish in many different combinations, they found that one specific combination was not compatible with life. And implying, therefore, that it is not either the nuclear genome, sorry, the mitochondrial or the nuclear genome that are sort of pathological, but it is their specific combination. And in this paper, they explained this with the fact that these 2 genomes had mutations, variants in residues that specifically interacted with each other. And these interactions were disrupted, therefore showing that mitonuclear incompatibilities have a role in evolution. And these 2 genomes interact through their gene products. So new mutations in the mitochondrial genome need to exist in a heteroplasmic state. What does heteroplasmy mean? Heteroplasmy means when within a single cell you have different copies or different versions of the same DNA molecule, mitochondrial in this case. Like in this case, this is the 3243 mutation, which is the most common mutation found in humans. And this mutation is in a tRNA gene. And this concept of heteroplasmy is very important. It will be important in this presentation because it modulates the effective penetrance of the mutation. So if the mutation is present, unless it is present at a high enough level, 60 or above, or 70, depends on the context, there is no pathogenic effect. You have to overcome the biochemical threshold. But once you overcome this biochemical threshold, these mitochondrial mutations also have an important role in human disease. So as I said, this 3243 point mutation can cause really severe phenotypes in humans, a wide range of phenotypes. But this is not the only type of mitochondrial disease. So this is a plot that shows the different sort of roles of mitochondrial mutations in disease. And you have cases like MELAS, where the mutations are very rare in the population, but these are very severe. You have sort of midway variants that are not very penetrant, depend on the nuclear background. And then you have variants that are found in sort of populations that can confer, at homoplasmic state, that can confer susceptibility, increase susceptibility to certain common diseases. So recapping over the introduction, we have the eukaryotic cell, and especially animal cells, are made out of 2 genomes, and the mitochondrial genome mutates a lot faster, challenging homeostasis in development, inheritance, and aging. But there is an upside to this, that the mitochondrial genome is actually cleared maternally, as in when the genome is transmitted to the offspring from the mother, there is a purifying selection, which means that the mutation is decreased. There's less of it or disappears. So this purifying selection has been mostly sort of assigned to the mtDNA bottleneck. These are a lot of evolutionary terms that we apply to sort of more cell biology sort of concept, but the bottleneck means that there's a decrease, like in populations, there's a decrease in number of copies of mitochondrial DNA in the developing female germline. And this transient increase exposes mutations that perhaps are at levels that wouldn't be detected below the biochemical threshold, exposes them to segregation, meaning that they can decrease a lot, but they can also increase a lot. And this exposes new mutations from one generation to the other to selection at the cellular organismal level, meaning that new mutations, even if they come out at very low levels, they can only in one generation disappear because they're not compatible with life. So the germline is actually a context of exceptional quality control for mitochondrial DNA. We have the bottleneck in pre-emergent germ cells, but emerging evidence shows that even in oocytes, there is a lot of quality control. But the question that still remains to be answered is there a subcellular quality control system for mitonuclear incompatibilities in the germline? So we know, for example, that mitochondrial quality control is, and in specific mtDNA quality control happens in many ways. So we have the ubiquitin-proteasome system that was so wonderfully introduced by Jane. When the ubiquitin-proteasome system tags outer mitochondrial proteins with a lot of ubiquitin chains, this can initiate a cascade of mitophagy, whereby the damaged mitochondria are degraded by recycling. But there's also proteases that have been shown to remove mitochondrial mutations. There's mitochondria-derived vesicles that don't happen through the autophagy system, but happen through a transfer system that deliver mitochondrial DNA to the lysosome. And even more recently, there's a discovery of direct transfer from the inner mitochondrial membrane directly to the lysosome of oxidized mitochondrial DNA through pores. So the question is, do any of these, and if so, which are responsible for germline purifying selection? So to answer this question, we employed a mouse line that carries a heteroplasmic mitochondrial DNA mutation that is inherited. This mutation is in the tRNA, like we saw in common human diseases. And in specific, this mutation is in the same position as a human mutation that causes myopathy. So this mutation, and this plot is very important because I'll be using it later, and it's sort of the raw data to look at inheritance of mutation. This plot shows the mother heteroplasmies for one specific cross and their offspring. So each dot is a combination of mother to offspring. What this means is that when a mother had 58%, like in this case, the offspring inherited, in this specific case, about 10% of the mutation. And so what you can see, as I said, because of the bottleneck, when a mother has a specific heteroplasmy, like 60%, the offspring can inherit a whole wide range of levels of the mutation. But in the case of this mutation, there is a hard threshold at 80%, meaning that when you have more than 80% of this mutation, the offspring are not born at all, meaning that this mutation is either incompatible at very high levels with life, or it is purified. This mutation is quite a good model to study selection because in the germline, because it has even another layer of sort of quality control. So when mothers have high levels of the mutation, so above the threshold, above 60, the offspring tend to inherit, on average, less of the mutation, as you can see this trend line. And this actually models the most common human mutations, like the 8344 A2G or the 3243 A2G. And so, to try and figure out which quality control system might be involved in the germline selection, we took embryos at E8.5, which is really early at organogenesis, where each one of these cells can be sequenced in a single run for their entire transcriptome. And therefore, we ran an unbiased transcriptome analysis of all the cells in the embryo. And what we found is that there were a vast number of pathways were modified, but the top ones were these. And out of these, I noticed the ubiquitin-mediated proteolysis. So then we sought to, I don't need to explain this because of the introduction that was wonderfully made. And but we went further to sought to validate this effect. So we took mice from adult brains from mice, and we ran unbiased TMT proteomics. And what we saw is that there was a mild but sort of a signal of decrease of the proteasome subunits and the same ubiquitin-mediated proteolysis protein. This is a KEGG pathway that we used for the transcriptome. So we saw that there's a decrease in expression of UPS proteins, but we wanted to validate whether this was actually effective. So we made mouse embryonic fibroblasts from these embryos carrying the mutation, and then we subcloned them to have two populations, one carrying a lot of the mutation, one carrying little, with the same nuclear background. And on these cells, we performed RNA-seq, and this sort of showed the same pathway as the in vivo proteomics, where basically these are all the differentially expressed genes. We don't need to particularly look at them, but the kind of point is the general trend that there are more ubiquitin-mediated proteolysis genes that are downregulated, and that there are some proteasome genes that are downregulated with no compensatory upregulation. So then, using these maps, we actually sought to validate this effect further at a functional level. So these are cells, the same cells that I just spoke about, carrying the different levels of the mutation, and we stained them for mitochondria, which you can see here is in a network that is characteristic of this organelle. And then we stained them, and this is in green, and we stained them also for ubiquitin, conjugated ubiquitin. And then we looked at these spots where they localize, or rather how many of these spots co-localize next to mitochondria. So what you can see is that at low heteroplasmy, you have spots that co-localize next to mitochondria, showing that there is quality control of mitochondria at low heteroplasmy, but this is inhibited at high heteroplasmy. And then this sort of is even more pronounced when cells have been treated with an inhibitor of the lysosome, which blocks the flux and therefore allows you to measure all the mitochondria that were ubiquitinated and would have been degraded, and same with the proteasome. And then quantification done on multiple biological repeats showed that this difference was highly significant. So we've seen that this mutation impairs the ubiquitination of mitochondria, but this doesn't necessarily mean that this allows the mitochondrial. So the ubiquitination is how the mutation maintains itself. To be able to prove this concept, we need to restore mitochondrial ubiquitin. And so to do that, we employed this deubiquitinase. This deubiquitinase called the USP30. And this USP30 sits on the outer mitochondrial membrane and takes away ubiquitin tags onto outer mitochondrial membranes, therefore restraining excessive mitophagy. So to compare the inheritance pattern with or without USP30, we had to design a very specific cross. So the two parents here, you can see, are heterozygous for USP30. USP30 is a nuclear gene, so it follows Mendelian inheritance, while the mitochondrial is inherited only from the mother. And from the same mother carrying the same mutation, we can compare the inheritance of the mitochondrial mutation in the three sort of genotypes. But before showing you the data, I want to sort of show that this technique is actually very reproducible. So this is my data that I've gathered over a long time within this animal facility, and this faithfully reproduces previously published data that was gathered in different animal facilities over a different amount of time, showing that basically the threshold is at 60%. So with the threshold 60%, we then compared the inheritance of the mutation when the USP30 knockout allele was present, or rather USP30 was missing. And what we see is that the USP30 knockout allele does effectively modify the inheritance of the mutation. And we did a statistical comparison of this, and this was a very strong significance. So, okay, the USP30 modulates their heteroplasmy inheritance, but does it decrease the amount of mutation, or does it increase at this level? Because what happens above the threshold and what happens below the threshold are two very different biological phenomena. So we split this plot and pull together all the data from all the mice, all the inheritance points at below 60% of heteroplasmy or above. And what we can see here is that, as we know, this mutation, when mothers have high heteroplasmy, is inherited less by the offspring. But if you don't have the USP30 allele, this is inherited even less, therefore showing that USP30 knockout allele enhances purifying selection. So we went one step further. Beyond proving the fact that this did not kill the animals, it did not affect the health of the animals, this just decreased the amount of mutation inherited, we wanted to see at what stage in development. How can we do that? To be able to do that, you need to understand how the expression in the germline works. So these are the cells of the germline. This is sort of the genome is maintained here across the mother to offspring across the evolutionary timescales. And the PGCs and the oocytes express the same genes and the same alleles as the mother. But once the oocyte is ovulated and it gets fertilized by a sperm, there is a very unique event that happens, which is the maternal zygotic transition, when the maternal RNA and protein are degraded and they are to be replaced by the embryonic RNA and protein. And therefore, the paternal genes are only expressed from the maternal zygotic transition onwards. So we use this concept and we combined it with the fact with crossing the mice in different ways to dissect when developmental timing of selection of mutational removal. So for example, here the allele only comes from the father, meaning that it's only expressed after the maternal zygotic transition, and so on. So the first comparison we did was to compare these offspring whose mother was USP30 heterozygous, so their PGCs and oocytes were heterozygous, and we compared it to the inheritance of mother pair offspring where the knockout allele was not present at all. And what we can see is there's no significant difference, meaning that having a knockout allele only in the maternal cell germline is not sufficient. But then we went one step further. We compared it with the USP30 knockout offspring that inherited strictly from the father. And this also showed the same, that the paternal knockout is not sufficient. And therefore, this means that the knockout allele needs to be present in the mother, but also needs to be present in the offspring. And then we went one whole additional step to show that if the mother is knockout and not heterozygous, the offspring inherit even less. So when the mother is USP30 knockout, the offspring inherit less of the mutation than when the mother is heterozygous, showing that every USP30 knockout allele, when it's maternally inherited, confers additional removal, additional selection. And this therefore proves that the purifying selection or the removal of the mutation via the ubiquitin-proteasome system happens specifically in the maternal zygotic transition, as the maternal gene products are replaced by the embryonic. But we wanted to prove this in another way to further strengthen, because we're very skeptical about our own data. And we did this by assembling single-cell RNA-seq, public single-cell RNA-seq data sets of the entire germline. Thereby constructed an atlas of gene expression. And using this atlas, we could see that the USP30 expression, and you can look at these rhombus dots, is actually predominantly high in the early maternal zygotic transition, and therefore its loss will mostly affect this, or primarily affected the maternal zygotic transition, independently confirming our phenomenon. Using this atlas, we also showed, and a very interesting fact for the quality mitophagy field is that in the germline, the mitophagy genes and the autophagy genes and the ubiquitin metabolism, all these quality control systems are uniquely upregulated in the early embryo, and they're actually shut off in the oocytes. Therefore, further proving that the selection or removal by ubiquitin-mediated mitophagy must be happening at this step. Interestingly, the fact that selection acts at this step means that we have an ideal therapeutic window for treatment, because mothers who carry the mutation want to generally, and it's advised in a sort of clinical practice, for them to have in vitro fertilization. And therefore, combining in vitro fertilization with commercial USP30 inhibitors that are already in clinical trial could decrease mutant load below the biochemical threshold. And therefore, this is a new way to treat and prevent mtDNA disorders that affect 1 in 8,000 people. And so lastly, unfortunately, I don't have time to talk about the entire mechanism that is in my paper. But basically, what we show on a cellular level is that the mitochondrial mutation disrupts mitochondrial translation. This is relayed outside the cell, so global translation is slowed down, and degradation needs to adapt it as well. And this is the cause for the effect of the transcriptional slowdown. And because of this, then this means that there's less degradation of mitochondria. There is more, which means that there are more copies of mitochondrial DNA. And even though a lot of it is mutant, enough of them are wild type. And therefore, this effect allows the mutation to survive. explaining why some mutations are inherited, even though all these quality control mechanisms exist. And so with this, I'd like to thank you for listening, and I'd like to thank all my wonderful lab, without whom I couldn't have done any of this, and over all these years, and giving me a really wonderful time. So thank you for listening, and thank you for all the people that helped me in these years.
Thank you, Michele. That was really interesting, and thank you to you both for really interesting talks. We have some time for some questions now, so if you have a question, please pop it into the chat and specify who it's for. We already have some there from Jane's talk, so I'll begin with those, if that's okay. So the first question we have is from Prahalad, and it says, if pseudoligase trims are catalytic activity lacking E3 ligases, does pseudoligase oncoprotein complexes enhance oncoprotein stability more than degrading them through the UPS?
Oh, I don't know. I don't know any examples of that. That's a really great question.
Quite a few questions, long question.
Yeah, it's a really interesting question. So if I'm understanding it correctly, you want to know whether if there was a pseudoligase bound to a substrate, whether it would actually stabilize the substrate, I think. Yeah, I'm not sure. I don't know any examples of it, but I think it would be a really interesting avenue to explore, because I think that there is, in the pseudoenzyme literature, there is this kind of idea of sequestering substrates. So then by the pseudoenzyme, which then blocks the access for the active one. So I would suggest that it would probably, I think it could happen, but I don't know any examples of it myself, but I think it would be a really cool thing to look at. Maybe some mass spec would help with that.
There is a follow-up question as well from the same person that says, can pseudoligase trims be re-engineered, or if a pseudoligase can enhance oncoprotein stability, is there an application for PROTACs for that these targets can have for the pseudoligase? Sorry,
say it again. So targeting a pseudoligase to a substrate could act as a.
Is there any application of a PROTAC in that scenario? I think is the question.
I think that would be really interesting, because I think in a way instead of thinking about targeted protein degradation, kind of targeted protein interactions, I think is a really interesting idea. I think we need to understand first a bit more about what they are doing. So for some of them, I think we don't really know what their function is. So, you know, for TRIM15, I don't think we know anything really about if it's not got a ubiquitination function, what is it doing? So I think we'd want to be a bit careful about targeting something, using it as part of a PROTAC if we're not sure exactly what it's doing. But I think for some of them, like TRIM2 and TRIM3, TRIM2 active, TRIM3 is not active. And it seems that TRIM3 blocks the activity of TRIM2. Something like that would be quite cool to try and use as part of a PROTAC. I think that'd be really cool.
Okay, thank you. And so I will ask one to Michele now. We have Ibrahim, who says, thank you for your presentation. He said, in your presentation, you mentioned that the 5024 mutation you have impairs mitochondrial ubiquitination. He asks, how did you confirm this? And he's wondering if, oops, sorry, I just moved this. Technical issue because I can't see the full question. 1 second. Let's make this full. So yeah, how did you confirm the mitochondrial ubiquitination there?
Well, in the ways that I showed, I mean, mostly in the fact that the entire, so we saw basically this change in gene expression. We saw this proteomic change, something that I hadn't seen before. Obviously, we saw that mitochondrial ubiquitination was decreased at a sort of like a functional level with the imaging that I showed. But one thing that I didn't have time to mention is that basically we saw as well that there was slower degradation of all proteins using a sort of Western blot and comparing the accumulation with MG132. We saw that when there's a lot of the mutation, there's just slower degradation, and this is probably because of the less of the proteasome that we have. which is sort of quite interesting because it shows how this mitochondrial variant, a single SNV, can modulate the entire metabolism of the cell, can slow down translation and slow down the entire degradation. And if you think about what Jane said at the beginning of all the things that ubiquitin-proteasome do, like how that can happen without messing up the whole cell, how the mice are still fine. It's I guess it's a very small change, but it's something that I think I would like to study further. Yes. It warrants further investigation, let's say.
Okay, great, thank you. Okay, so we've got some more trim questions. So we have here from Benjamin, good talk. For Jane, you mentioned there were some hints for evolutionary conservation for some of these inactive trim pseudoligases. Do their expression levels also change between tissues and/or development, or in response to different types of stress? Does this give any hint as to the regulatory role on other trims or in other signaling pathways?
I love that question. Yes, they're definitely throughout the family. They're expressed in different tissues. Talking about all of the trims, they're expressed in different tissues at different times. So, for example, some of them kind of peak during neuronal development, are only expressed in neurons at a certain point, and then they go down. So what are they doing? And that's a question I'm trying to work on at the moment. In terms of the pseudoligases, yes, some of them are expressed in certain sort of tissues, and they have kind of correlation. But I think the one that jumps to my mind is TRIM22 is only expressed in response to interferon. So it seems like there's something there with inflammation and infection with TRIM22, but it's always been assumed that it was to do with ubiquitination because it has this ring domain. So a lot of in-cell studies have been done where there's infection, and then you see a readout of some kind of protein getting ubiquitinated, and it was assumed that it was because TRIM22 was ubiquitinating it. But actually TRIM22 might be maybe acting further upstream, doing something else. As someone who's been working on ubiquitin, it seems funny to be suggesting anything but ubiquitin. But I think that's what it's doing. So I think that there is this kind of tissue, temporal, spatial regulation of these different trims, and very much case by case basis. And there's no kind of one size fits all for the whole family.
That makes sense. And there's another question here that says, thank you for the very interesting talk. And this one says, my question is, do you think it's worth looking at methylation or epigenetic changes playing a role in the pseudoligase expression, particularly between species? So it's kind of along the similar.
I think that was a really good idea. I've not done anything at all to do with that. I think that's a really nice idea. It'd be nice to see if there are some databases out there. Maybe someone's done some kind of sequencing already, especially in terms of infection, whether there's some kind of correlation there, and whether we can look at that through some already published data. I think would be really cool to try and understand when and where they're actually expressed. It might give us some more clues about function. Thank you.
Thanks. Okay, and then we have a question here for Michele, which was just what made you decide on USB 30 over other USBs like USB 35?
Well, it's sort of, well, it's because it's the mitochondria resident, the ubiquitinase. So there are some of them that can associate with mitochondria, but the thing about USB 30 is that it is on mitochondria the whole time. And also it's being used a lot in mouse models. So we know that its knockout, which is being used in clinical trials, we know that its knockout doesn't particularly affect like sort of the mouse health. And therefore it was the sort of ideal gene to knock out to do these sort of complicated crosses.
And I guess maybe I have a follow-up question on that as well, which is, do you have any intention to investigate perhaps the ligase that's behind the ubiquitination? Do you think it might be pink-1 parkin, or do you think it might be another ligase?
So I'm wondering whether you were one of my 4 reviewers, because all of them asked for this question.
No, I'm not. Sorry if I've triggered you.
No, of course not. I think it's a very relevant question. The thing is, so having looked a lot at the mitophagy field, I think that personally, I think that there's a promiscuous. There isn't a single E3 ligase for mitochondria, nor is there a single E3 ligase that at any point in time will ubiquitinate mitochondria, except for when you have really strong stresses like depolarization or something like that, where Parkin specifically goes, but which is why I speak about ubiquitin-dependent mitophagy rather than Parkin-mediated mitophagy. But because 4 reviewers asked us to do it, we did we tried in cell, because we repeated a lot of these experiments or in cellulo to validate the sort of mechanism, and we showed that this was autophagy dependent, and we could also show that this was PINK1 dependent, which sort of makes us think that it is Parkin that actually can remove these mitochondrial mutations. And it sort of makes sense because the developmental timing that we found, the maternal zygotic transition, is quite unique. And then there's a massive induction of autophagy in that step. And that is to remove paternal mitochondria and is to remove all the maternal stuff. So it sort of makes sense with all the literature that Parkin can remove mitochondria, but only in very, very unique cases. And but I still think there's a lot more to know and to figure out about this, because I think my other 3 ligases might be involved. Thank you for your question.
It's very interesting, very exciting. Yeah. Okay, so we are running out of time. Perhaps one last question, and there's so many questions being asked, so I think that that's something that could be continued online. But the last question, perhaps for Jane, would be: do you think monomeric trims may interact with other proteins and restore ubiquitinating activity?
Yes, I think that we can't rule it out. I think that there are some really nice examples of this elsewhere in ring domains. For example, BRCA1 and BARD1. So BRCA1 is an active E3 ligase and BARD1 is not, but BRCA1 needs BARD1 for its activity. So you have this kind of heterodimeric ring complex. And I think that it's possible that some of these rings these trim rings act maybe in that way. Can't rule out that they're not part of an active complex, if you know what I mean. So I think that would be a really nice thing to do. Again, kind of following up with some IP mass specs and having a look and seeing if you can find some interacting partners there, and seeing if there's, because it would be a very strong interaction, seeing if we can find if it's part of an active complex, maybe as a response to a stimulus, something like that. I think would be a really nice one. I think it's definitely an option. That could be then like a regulatory mechanism. You see it for kinases as well, where you have these activating pseudokinases that activate the real kinase.
Okay. Well, thank you so much both for sharing your really exciting data. That was really interesting. And I look forward to sort of seeing what comes next as well. So I think we've come to the end of the time for the questions for now. Thank you to everyone for attending today's seminar, and for the speakers for providing talks. So, you can continue the conversation online if you would like. You can follow @BiochemSoc on Blue Sky and X. The Biochemical Society welcome suggestions for future topics and speakers to feature in the Biochemistry Focus webinar series. If you have an idea for a webinar in 2025, they invite you to submit a proposal for an upcoming webinar, and you can find out more information about the webinars at www.biochemistry.org. For all of the upcoming webinars that are listed on the website as well, and if you've missed any of the 100+ webinars that have already been run as part of this series, or if you'd like to watch them again, please visit the website or the YouTube channel. The recording from today's webinar will also be available to watch within the next couple of weeks as well, so we can recap if we want to. And then finally, I want to highlight that it's more important than ever to stay connected and engage with your fellow molecular bioscientists. You can join the Biochemical Society's community of researchers and specialists and take advantage of key benefits, including discounted registration fees for society courses and meetings, exclusive access to a wide range of grants and bursaries, personal online access to 2 of their journals, and more. I know that when I was doing my PhD, Biochemical Society grants were great for conferences and getting funding for that. So you can visit their website to find out more. And with that, I will say thank you to everybody. Thank you to our speakers again, and have a lovely afternoon.
Thanks, Hannah.
Bye.
Thank you very much.
Bye.